The overall goal of this video is to provide an integrated set of methods to identify and experimentally validate genomic regulatory elements, so-called enhancers.
This method can help answer key questions in the transcriptional regulation of biological events, including enhancer selection and usage during cellular differentiation or in response to external stimuli. The main advantage of the presented workflow is that the protocol can be easily adapted and used in any cellular model systems.
I will demonstrate the steps of the procedure.
To begin, choose a candidate enhancer based on pre-existing ChIP-seq analysis. Use a genome browser to view the results of the desired ChIP-seq experiment and identify binding sites in the proximity of the gene of interest. Choose Define a Region of Interest to obtain a 200 to 400 base pair sequence of the selected genomic region and use the sequences for subsequent primer design. With standard molecular biology techniques, amplify and clone the selected genomic region into a reporter plasmid construct. To test enhancer activity, transfect the reporter constructs into mouse embryonic stem cells by first adding 200 microliters of 0.1% gelatin solution per well to 48 well plates. Incubate the plates for 30 minutes prior to plating. The day before transfection, plate feeder-free embryonic stem or ES cells in 250 microliters of ES medium at a density of 3 × 10⁴ cells.